Hi, everyone. Welcome to OsteoBites. Thanks so much for joining us today. My name is Christina Iptoma, and I am mom to Osteo Angel Dillon and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are joined by Dr. Amy Armstrong from Siteman Kids. She will discuss a new open-label, cohort-sequential, dose-escalation, and dose confirmation phase 1/2 clinical trial to evaluate the safety and efficacy of Domatinostat in combination with Sirolimus in adolescents and adults with relapsed refractory sarcoma and osteosarcoma. Thanks so much, Dr. Armstrong, for joining us on OsteoBites today. Everyone, please feel free to submit questions during the presentation using the Q&A feature in Zoom, and we will share those with Dr. Armstrong after her presentation. A little bit more about our guest today. Dr. Amy Armstrong is a pediatric oncologist who directs the solid tumor program at Siteman Kids, affiliated with St. Louis Children's Hospital, as well as co-directs the adolescent and young adult sarcoma program in collaboration with Siteman Kids and Siteman Cancer Center. She's an associate professor of pediatrics at Washington University in St. Louis and has research interests in neurofibromatosis type 1 related plexiform neurofibromas, renal tumors, and sarcomas found most commonly in the adolescent and young adult population. Dr. Armstrong serves as the site principal investigator for the Children's Oncology Group, Neurofibromatosis Clinical Trials Consortium, and the National Pediatric Cancer Foundation, and is invested in conducting and developing clinical trials to serve a diverse range of solid tumors in both the upfront and relapse setting. We are thrilled to have you on OsteoBites today, Dr. Armstrong. Thanks so much for joining us. I just have one announcement before we get started. We are in the last week of September, and that means it is still Childhood Cancer Awareness Month, and we'd love for you to go gold with us through our Gold Bow campaign. Each gold bowl is more than just a decoration. It's a symbol of hope, resilience, and solidarity with kids and families facing cancer. Together, we can turn neighborhoods gold and make a real difference, one bow at a time. You can get involved by making a donation to receive a bow or a pack of multiple bows, or you can sign up as a Bow Agent volunteer. Bow Agents help spread awareness by placing these beautiful gold bows around their communities, on mailboxes, storefronts, schools, and more. I'm going to put a link in the chat where you can get more information. And I know in my neighborhood, those gold bows stay up all year round. So it's not too late to participate and get started. So with that, let's go ahead and get started with our presentation today. Dr. Armstrong, if you want to go ahead and start sharing your slides.
Yeah, give me over. All right. Okay, here we go. Well, Christina, thank you very much for the kind introduction, and I hope you can see everything appropriately here. It's always confusing with the multiple computer screens that we have the right slides up there, but it sounds like we are good to go. But I really appreciate the invite today to be able to discuss our kind of handful title here, open label, but basically phase 1/2 study for relapsed refractory sarcomas and specifically osteosarcomas, exploring the HDAC inhibitor domatinostat in combination with sirolimus. So really a pleasure to be here today, and thank you for the kind introduction. Let's see, there we go. All right, so before I get started to the premise of the study, I wanted to first give a shout out to the consortium and collaborations that I've had the pleasure to work with over the past couple of years. So to provide a little bit of background about the National Pediatric Cancer Foundation, or the Sunshine Project, they are a philanthropic organization that now entails about 40 different sites scattered throughout the United States. Try to put a little bit of map here so you can see all the institutions that are involved. But it's a really rich opportunity to be able to conduct clinical trials in what it's otherwise known as just kind of rarer diagnoses. So being able to reach patients in different areas, to enroll quicker onto studies, and to really understand the activity of agents is a prime mission here. So an absolute pleasure to be able to work with them. And then the Sunshine Project, or the National Pediatric Cancer Foundation, has been working really closely with Vujade Sciences over the past couple of years, really to prepare for the launch of this phase 1/2 trial for children and adults with osteosarcoma. And then we were able to expand out to other sarcomas for the phase 1 portion. Just a shout out that most recently, Vujade joined forces with Helix, which is an AI-driven biotech focused on rare diseases. I've actually had a chance to work with Helix on some of my neurofibromatosis work as well, so it's been a nice marriage between the companies. But it's an exciting new relationship. Definitely want to highlight that Vujade changed its name over to Helix USA. And so that's the verbiage that we use kind of throughout the study and within our protocol. I'm so excited to continue to work on this collaboration, really to see all the stuff that we have moving forward. So moving over to osteosarcoma, which is really the premise of why we designed what we've done and what Vujade or Helix had been working on over the past couple of years, if not longer. I don't think I need to tell you guys that osteosarcoma is a really difficult, albeit rare, diagnosis that we see, and we are in desperate need of more therapies and more options for patients here. We know that it's the most common primary malignant tumor of bone, occurring primarily in children and adolescents and young adults. There's a little bit of a bimodal age distribution. Obviously, older adults can get this as well. But what's unfortunate about it is really since the 1980s, we haven't made much more advancements in our treatment approach for osteosarcoma. So it was great when we learned that using chemotherapy in addition to surgical resection of the tumor would improve survival and reduce event-free survival, of course, or increase event-free survival or reduce events. But since we've realized that, we haven't made a lot of motion. And it's unfortunate when I'm teaching our fellows or we have new patients come through that, yeah, our standard of care in the US is MAP chemotherapy, methotrexate, doxorubicin, also known as adriamycin and cisplatin. Also, ifosfamide is known to be very active and in some regimens as well. But that therapeutic approach is the same as it was a couple of decades ago that we're using today. Through the Children's Oncology Group, we're trying to understand if adding a TKI to upfront therapy, especially in the metastatic population, will improve outcomes. But we really haven't made major advancements, and the goal is to see what we can do moving forward. And it's amazing that we have an organization here with patients and families and researchers and clinicians, really to fuel the fire for how we can move things forward. I think the really interesting thing about osteosarcoma and, a variety of other sarcomas and malignancies as well is, what will cause a problem within the disease. So the vast majority of osteosarcomas are going to be localized up front. There is a portion, obviously, that will be metastatic, most primarily to the lungs. But for the localized portion, if you just resect the tumor, that's not going to be enough. We've known that amputation and, back earlier in the 1900s, mid-19th 20th century, that amputation or surgery alone would eventually lead to tumor recurrence, most commonly in the lungs, and then that's where we were able to advance outcomes using chemotherapy. What's really interesting, though, is that we don't know of this other areas of disease up front. And so there's the thought that there's a metastatic recurrence or that there is micrometastases or small tumor cells that we can't see on imaging that will eventually cause a problem, reside in a lung niche, and a lot of researchers throughout our nation and internationally are really focused on how to work with that type of tumor microenvironment. We know that lung metastases can be resectable. In my opinion, I feel like not every osteosarcoma patient is the same. So upfront versus, those who relapse or have refractory disease. Those who have a relapse 2 years out and maybe one single lesion are going to behave differently than those who relapse a lot shorter in or with multiple sites. Can we cherry pick and remove tumor and then see how patients do versus patients that need additional treatment? So just like, all of us, not every single osteosarcoma diagnosis is the same, and really trying to tailor approach and treatment is extremely important. What we have been able to learn, though, throughout all these trials and trying to advance what we're doing is what we expect to see for patients. And so for patients who do have relapse and do have measurable disease, when our trials are inactive or we don't see response from the therapy, what do we expect to see at 4 months? And I think kind of understanding some of those historical controls, and also for patients who have had relapse and then their relapse was resected and they have no evidence of disease, what do we expect to see at a year? So we've at least been able to acquire that data, which really serves as the benchmark as we try to move other agents forward. All right. Perfect. So I think for osteosarcoma progression, what's really important to note is that recurrence is frequent despite complete removal of the primary tumor, which I just spoke about, and really enables us to focus on the micrometastatic disease and anti-metastatic efficacy. And the group Helix now really did a lot of work, which we'll talk about what metastatic endurance is. So how does a tumor cell remain and evade upfront treatment, evade, surgical control, evade chemotherapy, lay dormant for a while, but eventually cause problems. And that's really the niche that we're trying to focus on here and how we're trying to help people moving forward. All right. And so how can we address this problem and, really create a unique biological target is the premise for the study that we're about to run. And hopefully I'll work through some of their data well. So if you have any questions, I'll make sure I direct it to the right people. I was not involved in this preclinical work, but definitely believe in the science as we're moving forward into a clinical trial. So what exactly is this metastatic endurance? We feel like it's a unique phenotype involving stress adaptation, survival, and dormancy. So again, a tumor cell is able to remain dormant, is able to withstand the stresses of the environment, obviously is not going to be, completely remove a cytotoxic chemo, but will potentially reside in lung parenchyma, lung tissue, and eventually cause problems where we can see evidence of like pulmonary nodules on follow-up scans. And really kind of enabling these micrometastases to survive for months or even sometimes years in osteosarcoma patients where you otherwise thought the patient was in remission. Just try to move the slide. Hold on. There we go. Perfect. And so really the occurrence of cancer years after the initial treatment may arise from these disseminated tumor cells, which have colonized to distant organs such as the lungs and remain dormant or quiescent. Really, these dormant kind of disseminated tumor cells, again, don't respond to the initial upfront treatment approach. So the MAP chemotherapy that a patient received wouldn't eradicate the cell type, but the cell type also wouldn't be aggressive enough to where you were seeing it up front or seeing evidence of it up front throughout your initial treatment. And a lot of our cancer drug development is really directed at killing rapidly dividing cells, inducing apoptosis, being extremely cytotoxic, not necessarily cytostatic. And so are we really addressing the unique biology that we can see in kind of metastatic recurrence or recurrence of sarcomas and then even osteosarcoma here. And so looking at the goal of how do we target metastatic endurance, how do we target these dormant tumor cells that remain and have evaded the upfront treatment approach. The goal is really to achieve sustained therapeutic impact by disabling the cell's metastatic endurance to result in a substantial, durable clinical benefit for patients. Key outcomes are very simplistic. So we want to prevent recurrence and progression. Will this approach help patients who are otherwise in remission? That was the goal of kind of the drug screen and what we were looking at up front. Can we increase overall survival? Can we improve quality of life? These are all really important things. And obviously, as a pediatric oncologist working day in and day out and seeing how cancer impacts so many different people in so many different ways, how can we really extend but really brighten the life of our patients and families? So how can we go about this? So de-risk drug combinations, how can we leverage really a cost-effective, relatively rapid combination development and agents with known safety, tolerability, and pharmacology? I think one of the more frustrating things for those of us who are clinical trialists or translational researchers is getting access to drugs, having people buy into what we want to use the drugs for. Drug could be marketed in maybe a more common type of cancer, maybe more commonly in adults. Maybe it lost some of its activity and no longer are we able to look at that drug for a pediatric or adolescent type of cancer. So it can be a little bit frustrating in trying to get access to drugs and trying to make sure what we are using has some data behind it to be able to more easily translate it. And then target multiple broad and complementary cancer networks. We know cancer's smart. I don't think I have to tell the group this. We know, again, these cells may remain dormant and are evading upfront treatment. So how can we really hone in on these different networks that are at play and interplay throughout the weeks to months, after treatment ends? Can we increase efficacy? Can we address tumor heterogeneity? Obviously, a huge topic. Not every part of the tumor, I believe, is the same as another part. And really overcome acquired resistance to a single agent therapy. And so here, hopefully I'll be able to take you through the slide and the pictures, but really what Helix did was they used an adaptation of a previously reported three-dimensional reconstituted basement membrane extract, or 3D BME system that modeled breast cancer, initially tumor dormancy. And so breast cancer cells remaining dormant and then have eventual outgrowth. And so they're modeling this to model metastatic endurance. And they used it with low and high metastatic osteosarcoma cells. And this really allowed for the expansion of screening assays to then move into more sophisticated modeling systems where they use the PUMA approach, or pulmonary metastasis assay, which models lung metastatic niche. Then the 3D basement membrane extract really became important for kind of a high throughput modeling system to then be able to study what combinations or drugs that we felt would be helpful. And really based on this screening platform, Helix identified a combination therapy which consisted of sirolimus and domatinostat as promising for clinical evaluation in metastatic osteosarcoma. And so mTOR inhibitors such as rapamycin or temsirolimus, you can see here, and HDAC inhibitors such as the domatinostat, which is a selective class 1 inhibitor, were found to be really highly active and selective against conditions of metastatic endurance. And you can see how these are to the upper right-hand side of that bottom graph there with the rapamycin, domatinostat, and temsirolimus. Again, temsirolimus is another type of mTOR inhibitor, but really selective. And I think what's really interesting, though, is that agents like cabozantinib or regorafenib or saracatinib, which are known TKIs with activity in osteosarcoma, so that's been reported in clinical trials in the relapsed refractory settings, they're showing up here, and they're showing some mild selectivity to metastatic endurance in this screen. And then even more notably, methotrexate and doxorubicin, right, are agents that have been around for years and years and years, part of our upfront chemo backbone here. They were selective and active against highly metastatic osteosarcoma cell lines. So they were active, but they were not selective for the conditions of metastatic endurance. So while they're very cytotoxic and helpful up front, maybe not as helpful with this metastatic endurance phenomenon. And then cisplatin, which is the other agent in the upfront chemo backbone here, was active against highly metastatic osteosarcoma cells in the 2D as well as the 3D assays, but only in a very small micromolar range. And so in general, what I think we're learning here is that there's some targets that maybe we didn't quite think of, that this seems to me as a clinician who is obviously trying to help patients in the clinic, that the other drugs that are popping up here definitely have activity. And to me, that better supports kind of the approach that we're looking at here when we actually feel like rapamycin, domatinostat, temsirolimus could be really helpful for selectivity against the metastatic endurance here. All right. And so moving forward with the data, and again, it's a lot of preclinical stuff, so I hope I don't glaze over too many eyes, but I'll get to the trial premise soon. But data for the in vitro 3D basement membrane extract assay with this highly metastatic osteosarcoma cell line treated with sirolimus, domatinostat, or the combination is what we're seeing kind of in the first graph there. And really the highly metastatic cells treated with the combination of sirolimus and domatinostat led to a really appreciable inhibition of 3D growth beyond the inhibition expected by either drug alone, then obviously better than the vehicle control or no drug at all. Then as you move over to the right-hand side of the slide, you'll see that the combo of domatinostat and sirolimus led to strong inhibition correlation with loss of GFP signal in the lung tissue itself, which really shows you % lung tumor burden. These are just kind of representative images of the lung slices. So treated with vehicle control, you're super bright. Treated with your combination of sirolimus and domatinostat, very dull. And that was shown on the right here. Let's see. Trying to move a slide. There we go. Sorry about that. So what do you know about domatinostat and sirolimus? So they pop up in a drug screen. They seem to be able to translate well over into this pulmonary metastatic model where we're really looking at activity in lung tissue. But what do we think would be helpful about these drugs or about this combination? And I will say that these drugs have been explored preclinically and clinically in a variety of cancers to date. But domatinostat is a selective class 1 inhibitor, which I'll explain kind of the difference between the other HDAC classes. It's known to be a regulator of epigenetic activity and also tumor microenvironment modulator. Domatinostat has been studied in clinical trials. It was generated by a company called 4SC. I think there are at least 6 clinical trials in which we have safety data, which is really helpful. talk about that a little bit more coming up. And that we personally, as our big group between Sunshine and Helix, have exclusive rights and access to the drug. And I'll tell you again, as a clinical trialist, that is huge, knowing that we have the drug in hand, that as we're trying to push through the science and push through the protocol, that we are able to move the drug into clinic and be able to actually help patients or try to help patients. Then if we move over to Sirolimus, this drug has been around the market for a long time. It's used in cancer, but it's also used in solid organ transplants to help with mediate like immune rejection. It's used in vascular anomalies patients. I mean, it's used for a variety of other indications where we have a better sense of activity. And we also have a better sense of side effects, but it is a very potent mTOR inhibitor. Again, FDA approved for a variety of other indications. And really, I'd say as a clinician, I'm pretty comfortable with managing it and the safety data at hand. All right. So we talked a little bit about the drug classes for Domatinostat and Sirolimus, clearly different classes. The goal is that we're really inhibiting and targeting different networks of cancer pathways to try to optimize response here. The primary mechanism for Domatinostat is to block histone deacetylation, opens chromatin, and reactivates tumor suppressors. Then for Sirolimus, as an mTOR inhibitor, it obviously inhibits mTOR signaling, blocks stress adaptation and growth reactivation. And then between Domatinostat and Sirolimus, we have other key molecular effects with histone acetylation, tumor suppression reactivation, oncogene expression. And then moving down with Domatinostat, really looking at the stress response, apoptosis, DNA repair, and protein degradation. Then Sirolimus with more metabolism, proteasome function, autophagy, and apoptosis. Then I'll lastly highlight that Domatinostat is a selective class 1 HDAC versus some of the class 2 or pan HDAC inhibitors. So it is a little bit different than some of the other drugs on market. We feel that, it has nuclear localization and the selectivity is higher versus a more challenging class 2 HDAC. And then really that therapeutic window is very favorable, supports systemic oncology use. Toxicity profile, better manageable in clinical settings. I think we have some evidence with use in other clinical trials for the safety data here. And the development priority is that a class 1 HDAC is going to be a preferred class for safe broad spectrum targeting here. And I'll add that Domatinostat has been used as monotherapy. So we really are understanding the safety data from the drug. And then it's also been used in combination mainly with immunotherapy in 6 different clinical trials in Europe. And so I feel like one of the studies that helped me a lot was a study called the TOPAZ study, which was for advanced hematologic malignancies. But understanding the safety profile there made me feel comfortable, moving this drug forward, and especially with the Sirolimus, as we're going to dose it a little bit lower and hopefully very well tolerated as we move forward. All right, so the combination is now kind of termed Helix 4310. And the therapeutic rationale for using this drug combination is that we're going to disrupt, at least we hope, key metastatic endurance functions in osteosarcomas, and really other malignancies. And then by targeting non-overlapping pathways, the goal is really to increase efficacy and reduce drug resistance. That we're going to address tumor heterogeneity. Again, not all parts of the tumor are the same across and within patients. Not 2 patients are the same. Hopefully achieve durable clinical responses, and then really have a favorable tolerability and safety profiles, obviously our goal. Additionally, I would argue that micromets are really under constant stress in the tumor microenvironment, and we're hoping that that will make them more vulnerable than rapidly dividing tumor cells to therapies that add further stress by targeting their strain survival mechanisms. So we really hope to selectively eliminate these cells or their ability to grow and reduce the risk of recurrence and metastases. And so that brings me to the introduction for this clinical trial. So again, it's going to be run through the National Pediatric Cancer Foundation, or Sunshine Project. Where we're at right now is getting kind of approval in our protocol through Moffitt in Florida, which is the hub where the Sunshine Project's located. All of our studies start through a Moffitt approval. So the first patients are usually enrolled at Moffitt, and then we expand out to all the 40 sites who would want to participate, and it goes through their IRBs. Christina was asking me at the beginning of this talk, when do we expect to be able to enroll? fingers crossed, maybe the end of this year versus Q1 2026. We really feel like we're almost there to be able to provide access to trialing this combination. So like most studies where, domatinostat has not been directly studied with sirolimus, and in these doses, we're going to start out with a phase 1 portion to really test the safety of this combination here. As doing so, we also recognize that there's really an unmet need for other sarcomas. As soon as you hit the relapse or refractory era of sarcoma, it can be very difficult to be cured. And so we wanted to open the door for all different types of sarcomas to be eligible for a phase 1 portion. We do feel like some of these pathways are very active in other sarcomas as well, like rhabdomyosarcoma. There's obviously the COG trial back in the day that showed activity with temsirolimus and then oralamine cyclophosphamide, temsirolimus being another mTOR inhibitor, potentially in combination with an HDAC. Would that be a benefit? And so we'll kind of see if there's any signal of activity that we're seeing here. But patients who would be eligible for the phase 1 portion would just have a recurrent or refractory sarcoma. And then we'll assess the safety and tolerability. And so we're looking at 3 dose levels to either reach our maximum tolerated dose or recommended phase 2 dose, where we're advancing the domatinostat, but we're going to keep the sirolimus constant. And so the goal of sirolimus is to dose it at 2 milligrams daily. We do have built-in sirolimus dose reduction for certain sirolimus-related toxicities, but the vast majority of what we're trying to do here is to understand the most therapeutic and I guess the safest combination for domatinostat in combination with the sirolimus. So the initial dose level 1 or 0 will be 100 milligrams daily. The next dose will be 200 milligrams daily, and then the last dose will be 400 milligrams daily, which will be divided twice daily. So 200 in the morning, 200 at night, which we know was tolerated in other clinical trials as well. And then that will be combined with the sirolimus 2 milligrams daily as well. There's a simple 3x3 design here. Ultimately, we'll have 12 patients, so we'll need a total of 6 patients to show us that a dose level is therapeutic. So if we're able to get to the highest dose level, that would be 12 patients who would be involved in the phase 1 portion. Then we also have built in a dose de-escalation for domatinostat if needed, where the domatinostat would be dosed every other day. All right, I know this slide gets a little worried, but for those of you who sometimes don't see the intricacies of clinical trials, really the goal of a phase 1 study is to figure out, is what I'm doing safe? Am I doing any harm to the patient? So what we do in that first cycle, which is 28 days, and I should add that these drugs are given continuously. So domatinostat, sirolimus every single day for 28 days. The domatinostat may be twice a day if that's ultimately the dose level that you would be on. And then the cycle is 28 days total. And then in those first 28 days, are we seeing any type of major toxicities or dose-limited toxicities that would ultimately impact how we would want to dose things moving forward? The DLT period is just for the phase 1 portion. Now, of course, if dose issues or toxicities exist at other times, we will address and modify the dose of the medication if you're outside of that 28-day period or if you're even at the phase 2 portion. But if a major toxicity develops, and I haven't gone through all the nuances of what that means, but we would hold both the domatinostat and sirolimus, make sure that things calm down well, and then we would resume the domatinostat mostly at a lower dose. Now, and the sirolimus at the same dose. If we feel like the toxicity is just sirolimus related, and there's 5 of those that I feel like are commonly known to be sirolimus toxicities, being a high blood sugar, high triglycerides, high cholesterol, really bad mucositis, or something called pneumonitis or inflammation on the lung tissue, then we would address the sirolimus at that time. Ultimately, we need to keep you on med for at least 21 out of a 28-day cycle. So we don't want longer than a 7-day treatment interruption. And for us to be having evaluable patients, we really want patients to be taking the medications at least 75% of the time. So we're really understanding the response here. All right. And then as we move on to the phase 2 portion, this will really focus on osteosarcoma. And I want to take a little bit of time to go through kind of the different arms that we're going to be addressing here. I will add, if we have other type of sarcomas on phase 1 where we're seeing some signal of activity, we will again try to maybe open a cohort or include that in the phase 2 portion as well. But specifically for osteosarcoma, which again is where the drug development for all this came about, we'll have 2 arms. The first is a patient who has a recurrence of lung disease from osteosarcoma, and then that disease is completely resected. So they enter the study with no evidence of disease or NED. And really the goal here is by putting these patients on Domatinostat and Sirolimus, are we controlling the metastatic endurance that we know can occur? And are we improving event-free survival? So we know from the LeMay study that really, at 12 months or so, for those patients who had a pulmonary relapse or resection, no evidence of disease, at 12 months, the event-free survival was about 20%. And so our goal is to see if we can get it to 40% by placing patients on this drug combination. And we expect about 24 patients to be enrolled in order to statistically get us to that point. Now, the phase 2 portion is going to be for patients who have osteosarcoma that is not fully resectable. So you have a relapse or refractory osteosarcoma, and you cannot go into a surgical remission. So you do have disease when you enter the study. For this patient population, we would want the patients to have measurable disease. So unfortunately, at least 1 centimeter of tumor, as seen on imaging, which helps us with RECIST criteria and just really overall evaluation here. And the goal for this arm B, is to improve our 4-month event-free survival from just 12% up to 40%. So we expect 16 patients here. The dose level that we would use would be the recommended phase 2 dose that we find from the phase 1 portion. And so if you look at the bottom of this diagram here, you have dose level 2. We anticipate being able to get to the highest dose level. Obviously, we'll see what happens, and then we'll break that dosing off to arm 1 and arm B. Now, when we met with the FDA, over a year ago, they really said, hey, you might feel like this is the best dose combination to use, but you might also want to think about if maybe 1 dose level lower could be as efficacious when you're going through here. So what we'll do as we're moving through is see if it's warranted to also open up another dose level, 1 dose level down, to include for our arm B patients, those who have non-resectable or measurable disease. So that's the gray cohort coming over time. But more or less what I want you to take from here is there are 2 ways to enter with osteosarcoma in the phase 2 portion. One is to enter with a relapse that's resected and then no evidence of disease, or to come in with measurable disease. And then the trial objective. Oh, yeah, of course.
On that.
Yeah.
Can patients for that non-resectable, can they have multifocal mets? Like, can they have, if they have bone mets in addition to lung mets, and that's unresectable?
Yes, they can definitely have multifocal mets. What I would add, though, is that some met needs to be measurable. And what's really annoying, I feel like, is bone mets alone are not going to be measurable. And so what's ever in the lung would have to be at least 1 centimeter in size, but they are definitely allowed to have bone mets as well.
Okay.
All right. Any other questions before I keep going?
Nope.
All right, got it. All right. Yeah, I can't see you, so I assume silence is the answer. So for the trial objectives and endpoints, the primary objective here for the phase 1 and 2 study is to evaluate the safety and tolerability of combining Domatinostat with Sirolimus for phase 1, relapsed refractory sarcomas, versus phase 2, which is relapsed or refractory osteosarcoma. In phase 1, to get to that maximum tolerated dose or recommended phase 2 dose. And then in phase 2, to actually look at that anti-tumor activity of Domatinostat and Sirolimus. Secondarily, we're going to look at PK and PD parameters for combining Domatinostat and Sirolimus, and then document any observed anti-tumor activity in subjects with other sarcomas, so not osteosarcoma. Then from an exploratory standpoint, assessing liquid biopsies to determine disease status here. And then how are we going to do that? Goes to our endpoints, which are pretty consistent between trials. So it shouldn't be anything new to people, but for phase 1 and 2, looking at safety, We're going to look at the frequency and severity of an adverse event. So every time you guys have an issue, whether it's a rash or nausea or vomiting, well, we have to grade that. So how bad is it? And we have really strict parameters that we follow through the CTC AE guidelines that helps us grade a toxicity and then know what to do about it. So we'll make sure that we report those, and then any serious adverse events or any events leading to hospitalizations or things like that. We would look at the frequency of dose interruptions and dose reductions, and then the nature of the DLTs in the phase 1 portion. Then also in phase 1, the PK parameters, so a couple of different measurements for that. I think this is really important. We're going to look at Sirolimus trough levels, but we're not going to have the Sirolimus trough levels guide our dosing for Sirolimus. So I know a lot of people that may be listening here may not be used to use therapy with Sirolimus, like everolimus, but described assays have been created that can give you what that level of drug is in the blood, and we use it for a variety of other indications. Here in this study, we're not going to be shooting for a certain trough level. I think I've learned from the SARC consortium with using Sirolimus on prior studies, and just in talking to some colleagues who are using it and understanding kind of the safety effects of Sirolimus versus the therapeutic benefits. Really, these trough levels don't correlate. So I'm not going to have patients adjusting their Sirolimus dose based on a trough level that I don't know is significant. We're going to adjust Sirolimus based on toxicities, so that we are very well versed to do. But it'll be helpful as we move forward that we kind of understand what those levels look like in the blood. And then for those patients with the measurable disease, we'll use the RECIST criteria. And then for those with non-evaluable disease, response will be evaluated as able by the study team. I'm going to the phase 2 portion. For Arm A, we'll look at event-free survival at 12 months in patients with relapsed osteosarcoma who achieve a surgical remission. And for Arm B, event-free survival at 4 months. So again, these are the patients that are coming in with disease. And we'll be able to see that for comparison purposes. We'll also look at the overall response rate at 4 months as determined by RECIST. Then lastly, for Arm A and B of the phase 2, overall survival, and then also looking at liquid biopsies. So kind of a wordy slide, but really trying to get to our aims of figuring out if things are safe and then if we're helping anybody. And these next 2 slides, I think, are really helpful for the audience who might be interested to understand the inclusion and exclusion criteria of studies. Now, I'll say these can be a little nuanced and can be frustrating at times if you meet everything but one inclusion criteria. The goal, again, is that we're making sure that things are safe and really available to the greater population. And so people who enter the study need to be able to be adequately evaluated for safety and then also adequately evaluated for efficacy as we move forward. So key inclusion here is that the patients have to be at least 12 years of age to 40 years of age. So I think that's a big one to write down. I put the weight on the exclusion criteria, but you need to be at least 40 kilos as well. So now we have some really small, younger kids. We need at least be 40 kilos, and we're able to use at least 12 years of age. And that's a pretty common cutoff right now used by the FDA. Now, if we're seeing activity or if we're seeing really good safety data, of course, we would think about expanding down to younger children, but this is just where we need to start. For the phase 1 portion, we're looking at any type of sarcoma. Patients may have either evaluable but not measurable disease per RECIST. They can have measurable disease, or they can have no evidence of disease at the last treatment intervention no longer than 6 weeks prior to enrollment. So if the investigator feels like it's in the best interest for their patient who's at really high risk of relapse to understand the safety of this combination, you would be eligible for the phase 1 portion of this study. For phase 2, we're looking for patients who have either no evidence of disease, they're in remission after surgical control, that's Arm A, or for patients in Arm B who have had unresectable but measurable disease per RECIST. Of course, patients had to at least undergone 1 line of standard of care systemic therapy, and this is extremely common. Obviously, we want you to get access to the things that we know are helpful, and that patients are not candidates for therapies available to those that are known to provide a survival advantage. And that your body is functioning reasonably well. So we want adequate bone marrow, liver, kidney, and lung function, and we have parameters in the protocol where those need to be. Specifically to the Sirolimus known toxicities, we want to make sure that the triglycerides coming in are at a reasonable level and cholesterol is at a reasonable level, because Sirolimus can modify that some, and that a random blood sugar is not too elevated, so not higher than 1.5 times the upper limit of normal. For the cardiac control, QTC is just kind of a measure of your cardiac rhythm a little bit. We want to make sure that's not overly prolonged because that can disrupt and lead to dysrhythmias, and that you obviously need to be able to swallow pills. Unfortunately, we don't have the Domatinostat in a liquid formulation here. Then for key exclusion,
Dr. Armstrong, sorry,
yeah,
one quick question on that
yeah.
slide. Can you just clarify the difference between evaluable versus measurable disease?
Absolutely. So measurable is a definition by RECIST where you have to have a lesion in the body that's not in the bone that is at least 1 centimeter in size. So essentially that you're able to measure the disease. Most commonly in a lot of these studies, and especially with osteosarcoma, that's probably going to be a lesion in the lung that's at least up to 1 centimeter. A valuable disease means you might have some evidence of disease that you can see, but it's not up to 1 centimeter in size, or it could be just located in the bone, which doesn't quite count for an area that would be measured. So a patient who has lung disease that's 6 millimeters, so 0.6 centimeters, that we feel is osteosarcoma, that would be a valuable disease. We're still able to follow that, but we can't quite measure it to the same criteria as used from a clinical trial RECIST standpoint. Does that help?
Okay, got it. Yeah, that's super helpful. Thank you.
Yep. Key exclusion, again, can't be too small, and that's just because we don't have safety data for HDAC inhibitors at really small weights yet. So hopefully we'll be able to move that direction with a little bit more activity or evidence here. Patients receiving other investigational agents, I'm sure you guys can understand that, or who are on agents for purposes of treating cancer. Known hypersensitivity to the agents, brain metastatic disease, unfortunately, would disqualify. We don't know about the blood-brain barrier penetration here for the drugs, so we want to make sure that we don't need to be penetrating that area. Major surgery within 2 weeks would disqualify from starting. However, if you wait a little bit longer, you'd be okay. History of a solid organ transplant. There's a lot of immunosuppression that happens after solid organ transplants. And I even told you, mTOR inhibitors could be part of that. So unfortunately, that would be a disqualifier. And then other medications that essentially would impact the drug levels within the body or the activity in the body. And so as you're a clinician or your provider, they should be able to screen for what these drugs are. But very commonly CYP3A4 inhibitors and inducers that are either moderate or strong. Herbal medications or inhibitors would be excluded here. All right, I promise we're almost there to the end to see if we have any other questions. But I think what's really fascinating here is the correlative science. And, I struggle when I'm in clinic and I am using scans still to tell people if they have no disease or if we're concerned about disease. The anxiety that goes into those scans, I know, is just extremely overwhelming. I'm trying to scrutinize things. Radiologist is trying to scrutinize things. So what we're really looking for is something called macrometastatic disease, right? Something that we can see with our eye that's going to be on imaging. The question is, what can we not see with our eye? That might tell us that there are tumor cells circulating around. There's really a push in a lot of different cancers, osteosarcoma, other sarcomas included, to try different methodologies for measurement that really help us understand, if a patient is truly in remission, if there's other levels of concern, things like that. I feel very fortunate to be able to work with Moss, who's out the University of Colorado. He's a part of the Sunshine Consortium and has agreed to work with us in the correlative science or biology feature of this study. So essentially what we're going to be doing is collecting circulating tumor DNA from arms A and B. So again, the patients with no evidence of disease and the patients that come in with measurable disease, and then also circulating tumor cells for the patients that come in with measurable disease from osteosarcoma to really be able to learn more about this disease. We have a similar trial where we're just really looking at biomarkers for a variety of other sarcomas right now that he's leading, and I feel like it's going extremely well. So happy and hopeful that we'll gain more data through the correlative science here. Additionally, we'll look at pharmacokinetics and pharmacodynamics, so blood levels of Domatinostat and Sirolimus, and then otherwise the modulation of known pathways for HDAC and mTORs. This, I don't know that we have any CRAs or anybody here on the call, but it's the infamous study calendar. I promise it's not that bad, and I'm not going to go through every detail. I'll hit the highlights on the next page. But to me, this is a pretty straightforward study. I don't think we're asking for too much. Some of this is going to be standard of care, pictures we would need to take in order to understand if disease is there or not, labs to make sure that everybody's safe, EKG or electrocardiograms to monitor heart activity every so often, and then our correlative science. So things that I feel like would otherwise be getting done in clinic, and then things obviously for the correlative science or advanced data that we're trying to achieve. And so the key points here are that really the visits are going to be weekly for the first cycle, make sure everybody's safe in that DLT or phase 1 period. Then obviously as we expand out into the phase 2, still making sure we're all safe. And then bi-monthly for the second cycle, so coming in 2 weeks in and then 2 weeks in. And then from that point, it's just monthly. And it's monthly with oral meds. And I feel like that's really great when we think about tolerability and what patients are looking for. Imaging studies of primary metastatic sites are going to be done at screening prior to cycle 3. So after 2 cycles, and then every other odd cycle, which is a pretty common routine or regimen for screening. And then obviously if there's confirmed for relapse or progressive disease and follow-up. For the phase 1 and 2 patients with osteosarcoma, we'll do the circulating, the cell-free DNA and the circulating tumor cells, which we'll be collecting at screening at cycle 2, day 1, and then day 1 of all even treatment cycles, end of therapy and relapse progressive disease. I think the next bullet points, and they're not boring you too much, are just really important because they influence the clinic visit, is really the PK and the pharmacodynamic samples. And so we'll be collecting those on cycle 1, day 1, and cycle 2, day 1 for both PK and PD. I think what's really important is just the end goal. So pre-dose of the medications and then all the way up to 6 hours post-dose. And so hopefully that's a manageable thing to do in clinic to keep people outpatient, to be able to drive into clinic, kind of stay for a variety of blood samples, and then be able to go home same day. Additionally, for the phase 1 osteosarcoma and then all the phase 2 patients that are supposed to be osteosarcoma, we'll have tumor tissue collection at screening. And this does not mean like a new biopsy or a new tissue that we're collecting. But we would want to be able to evaluate that. And then patients who do undergo surgical resection during therapy or time of relapse disease, if available, we would love to have that tissue for analysis. Then lastly, the phase 1 only will do trough of the plasma Sirolimus levels at a variety of time points, which I explained will not dictate the dosing that you're receiving, but will just help us as we move forward. I think that was kind of the last, there we go, slide here. But I really want to end with some major acknowledgement. So I wouldn't be here today able to talk about this trial without the support of, I would say, first Helix, who came up with the science that we're now able to integrate into a very multi-institutional consortium-based clinical trial to really reach so many patients and their families. So a big shout out initially to Damon Reed, who is the head of the Sunshine Project and wrote me in a few years ago. So I really value that trust. Lars Wagner and Michael Isakov, who's really helped me with just protocol development as we move through. And then now Jonathan Metz, who's taken over that role a little bit, and Matteo Truckle. Both of these guys are now leading the Sunshine Consortium, doing a really, really great job as we move things forward. I don't think any of us would be here without Jess Gramilla, who's just absolutely instrumental in helping with clinical trial conduct and getting things forward. Kelsey Titus, and then lastly, Vivian Yin, who serves as our statistician here, helping with trial design and helps us understand the amount of patients we need to ultimately show benefit. Then from the Helix group, I've been working with David Scott and Richard for, a couple of years now. I added Chris Mortel as one of the clinical leads here from Helix. There are a lot of other people on the Helix team as well, but just very appreciative of all the help and collaboration, and obviously to our eventual patients and families and collaborating teams. Just really excited to be able to work with you all. And I will end it at that and potentially stop sharing if that's all right.
That sounds great. That was fabulous. Thank you so much, Dr. Armstrong. I'm so excited about this trial for several reasons that we kind of discussed. I mean, first of all, just accessibility to patients through this national network of sites. And because the combo is PO, that you can just take it orally, it also just makes it easier for patients. And then even the eligibility guidelines, because it's inclusive of, resectable, non-resectable, or even you noted if someone has had MAP has completed a MAP, but their oncologist thinks that they might be high risk, that they could potentially go on trial as almost like a maintenance therapy,
Mhm.
right?
Yeah, yeah,
Prevent.
for sure. And that'll be for the phase 1 portion. Obviously, when we get to phase 2, we would need evidence of, a relapse or some recurrence. But no, 100%. I think the science was built around those who might have a recurrence, like a metastatic recurrence, go into remission and how we can prevent that again. But there's such an unmet need for so many more patients. And so I feel like really trying to understand if we can see some clinical benefit or some activity was really a push that a lot of us had moving this forward. And then the one thing I didn't mention, which I think is awesome, is that for those who aren't located super close to one of the Sunshine sites, should that site be open and conduct the trial, we're going to provide some travel reimbursement and just help with being able to get there. So hopefully that will be able to help patients and then be
That is,
able to help patients access more help as well. Yeah.
yes. Yeah, that's great. Well, everyone, if you have any questions for Dr. Armstrong, please feel free to put them in the Q&A. It should be on your Zoom menu, and we'll make sure we share those with Dr. Armstrong. But in the meantime, I'll just kick off with some questions. So for the Domatinostat, you had mentioned that it has been studied in some clinical trials. Is it currently being used in other diseases, either FDA approved or off-label?
Yeah, sure. So the clinical trials it was studied in weren't osteosarcoma or sarcoma. For SC, who had created the compound, is not currently moving forward with the compound. So it's not active right now in clinical trials. There was definitely some activity seen, but I would say we don't know what the activity in osteosarcoma is going to be right now. Yep.
And then, so this is so interesting, like this concept of, the chemo is killing these kind of actively, these, rapidly dividing cells, but not really being able to address these dormant
Yeah.
cells. And so kind of bringing in these two other drugs. And so you had also mentioned one advantage was being able to address the tumor heterogeneity across, like within patient, across patients. But I'm wondering, like, theoretically, does this combo kind of, given the mechanism of action, work the same across all patients, regardless of their, genetic drivers? Or do you anticipate there are some patients that might respond better?
Yeah, I think there's so much we don't know. So do I anticipate that all patients are going to derive clinical benefit? No, I think that's just the nature of the beast here, right? That not all osteosarcoma is the same. So I do think that there, for whatever reason, are going to be some patients that behave differently. We hope that, by being able to do pharmacodynamics and understand like pathways that are truly inhibited, I think it's really great to have an mTOR pathway, which we know is active in osteo with, the HDAC network, which is doing so many other things. So I think that combination, I hope, will be extremely helpful for like resistance or like escape mechanisms for sure. But I don't fully know, to answer your question. I think the more we're able to see who can derive benefit, and then try to figure out from those patients that do, what is it about them that's helpful, I think the better. I personally obviously wouldn't molecularly screen, anybody to get into this type of study right now, just based on the nature of what we do know. But yeah, I'm excited to see kind of the clinical benefit rate and then see if we can dig even deeper into the science. And so many of us want to do that. And I think it's part of our discussions at a lot of our meetings. Like, let's say you're on drug and maybe you do go for a resection, what does that tissue show? Like what are we able to see from a mechanistic standpoint of how you were responding to the drugs that we were on? So I'm hoping it will help. I'm hoping we can obviously learn more, but I think the reality is not everyone's the same. So we'll have to see a response.
Yeah. Okay, so I know there are definitely challenges to testing agents in upfront setting versus in relapse disease, but do you think, and I know this is a very important first step, but do you think there might be any advantages or synergy to administering these drugs in like with MAP as opposed to after?
Yeah, definitely. I think the goal of these drugs is for the kind of the disseminated tumor cells that are dormant, right? We're trying to attack a different type of stress mechanism in which these cells have bygone, a cytotoxic approach, but are otherwise able to adapt and survive. So your question is, would this help it up front, or would this help this maybe more of in a maintenance setting? I don't fully know. I think what's interesting right now, and I'm hoping we gain data from our currently open COG study using a TKI cabozantinib, which also, multi-tyrosine kinase inhibitor, really impacting a lot of different cellular processes to upfront map. Like, what did that do for us? I'm really curious. We have a study looking at cabo as maintenance for some patients. Like, what can that do? I think the biggest bang for the buck would be for our patients who come in with measurable disease. If we're seeing reduction in tumor size with this, then that now we're like mechanistically thinking maybe it's not just these like dormant tumor cells that we can't see. Maybe we're having a different role as well. And so maybe even adding that in the upfront setting would be different too. So I think it's a great point. I could see a role for it, but I think first we have to see activity.
Yes, yes. Okay, so yeah. So some questions for patients that might be considering enrolling. You did talk about some of the toxicities, but just as a patient, like what are some common side effects they might experience that might affect their quality of life or just how they feel day to day?
Yeah, so I think common things we might see, which maybe they wouldn't appreciate, we might see a little bit of blood count abnormalities, a little bit of like electrolyte disturbances. I think the Domatinostat was pretty well tolerated. There was some like a little bit of nausea or GI side effects. Sirolimus in general is well tolerated, minus some of the mucositis that patients can develop. So I think when I think about cabo, which I adore, I love it as a drug, it can be a really nasty one too. To me, the HDAC mTOR combination, I'm actually hoping will be a bit more tolerable than some of our other TKIs out there. Not a lot of skin toxicity that we know of from these either.
All right, that's good. I love that you said that you adore.
Well, I mean, I use a lot. I'm not, against it by any way, shape,
Yeah,
or form, but some
I,
people have
I
a really hard time
definitely.
tolerating it. Yeah.
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 Yeah. Okay. And then some other like kind of practical questions about. So you talked about surgery. I think was it like 2 weeks or something? 2 weeks washout from
Yeah. You have to be
surgery.
at least 2 weeks out from surgery because, yes, from a major
And what
surgery.
about radiation therapy? Is there a washout for radiation
There
therapy?
is, and I want to say it's like 6 weeks, but I would have to double check. It's the normal clinical criteria from radiation would be in there. Yeah, I can pull that up real quick if that helps.
Okay. Great. And then so sounds like more information about opening end of this year or Q1 next year. First site will likely be Moffitt. And for any clinicians or patients that are interested, what's the best way for them to get in touch?
Yeah,
Get
that's
more
great.
info.
I didn't quite put an email address on here, but I can easily like provide my email and then I think Dress Camilla, like I tried to highlight just how instrumental she is, but I think really just Googling the Sunshine Consortium, there should be contact information on the website. I think that would be the best avenue to understand which sites are actually open at that point. We all know that regulatory can take a while and each site's a little bit different. Where can you enroll? I think that the Sunshine Consortium website would probably be the best spot for more information.
Okay, great. And that is again fabulous that there's travel support
Yeah.
for patients that are interested in participating. That's I think really makes a huge difference for again like patients actually being able to participate and enroll in a trial. So that's wonderful. Well, thank you so much. This was a great overview of a very exciting trial, and we really appreciate your joining us on OsteoBites today and making it better for pediatric cancer patients and osteosarcoma patients. More information on this and all OsteoBites can be found on our website at mibagents.org, on our YouTube channel, and at your favorite podcast place. And we hope you can join us next week. We're going to be joined by Dr. Alana Church. She's a pathologist from Boston Children's Hospital. And she's going to discuss insights and highlights from the AACR Special Conference in Cancer Research, Discovery, and Innovation in Pediatric Cancer, from Biology to Breakthrough Therapies. That conference is actually starting today in Boston. So if you're not attending the conference this week, it's a great way to get an executive summary from one of the conference chairs. You can find our OsteoBites lineup for the next few months on our website. You can subscribe to our YouTube channel so that you never miss an episode. And if you have ideas for future topics that you'd like to hear about, please feel free to email us at events@mibagents.org. Thank you again so much, Dr. Armstrong, for spending time with us today. And thanks to all of you for taking time out of your day to join us. We hope to see you back here on OsteoBites next week when we chat with Dr. Alana Church. Thanks so much.
Bye. Have a good day, everybody. Bye.
Thank you.